metastasis, which is just referring to how the cancer spread. Now we can put it in this kind of kinematics where it goes from invasion. So tumor cells, from a tumor, cancer cells goes to invade into different area. And then we go into intravasation, so which is just really going into into the vessel from a tissue, and then it gets transported to somewhere, and then we go to extravasation, so it's getting out of the vessel, and then after that they start colonization, then form a tumor, and then angiogenesis is just the formation of a new blood vessel. So this table shows how certain type of cancer we see here on this column: breast cancer, prostate cancer, lung cancers, are likely to go to different organs such as bone, lung, liver, and brain, and peritoneum. The term here, the organotropism. So tropism is referring to what being attracted to stuff. So like phototropism of the plants, how like when you grow plants in the dark with just a shed of light, the plants tends to grow to the light, right? So organotropism is it's the cancer's tendency to attracted to or to go towards to certain type of organs. We see that for breast cancers, they are likely to go to bone and lung, rather than liver, brain, or peritoneum. For example, and prostate, for example, prostate cancers are likely to metastasize to a bone cancer or to bone, which is 90%, rather than brain, which is just 1.6% of the case. Now, one want to ask, so why would certain cancer prefer to spread to certain specific type of organs? And two theories is one is the distance, the one that's closer. Now the other theory is the type of tissues that they are similar tissues, I guess. So this is the mechanisms of the metastasis. So monoclonal. So what this is showing, we have this primary tumor, and different colors are representing different type of tumor cells. And when they circulate, so maybe this pink one goes to circulate and then become a secondary tumor. And so this is called a monoclonal linear, and then polyclonal branched. Two different type of tumor cells, they circulate and then start forming more of them. Polyclonal linear, as you see. Just two different types of cancers make up a secondary tumor cells that are made up of two. And polyclonal branched, when they are circulating, they developed more mutation in the process of it. And so when they're actually seeded into a secondary tumor, they're just composed of totally new or the mutated version of the tumor cells that is not present in the primary tumor cells. So base membrane is a acellular structure, so there's no living cell, but it's as you see over here, and they're just encapsulating the structure, and which is exhibited in muscles, testes, lens, our GI tract, etc. So what their role is basically a border, and for cancers they need to penetrate through this border to metastasize and to go to other places. And how they metastasize is, for them to do so, they have to go through EMT, which stands for epithelial mesenchymal transition. So they have to go from their epithelial states cells and turn themselves into mesenchymal cells in order for them to move. And that's basically comparable to epithelial cells being nice and settled with each others, and they are now trying to become a solo adventurer that's trying to go to different places, which represent mesenchymal cells. And one may ask, why do we even have this EMT genes, right? If it's only going to just cause cancer, and we're going to answer that question later. But coming back to EMT, so what this graph or chart is showing, so the GFP is really representing the cells itself. So ignore that. And so but up here it's representing mesenchymal cells. And then K14 is representing a multiplying epithelial cells. So from their epithelial tumor cells, which is more present here, the more red, and as they develop into more mesenchymal tumor cell, we show the lack of epithelial cells, and they all turned into mesenchymal tumor cells. And here, similarly, red is representing, or I guess in the other way, red is representing a mesenchymal cells, and green is just the epithelial cells. So the GFP is really just showing this just the cells, and the red, and we just see more red. And mesenchymal cells, they do not reply, and it's only just to like trying to look. They're only just trying to look for places. So as shown here, and the epithelial cells, there's a high rate of proliferation, and when you go to mesenchymal tumor cells, there's barely no proliferation. But what they are high in is the invasion, how great they can like invade, whereas it's the exact opposite for the epithelial cells, where they can't really invade well. So EMT is characterized by the loss in cell polarity. Polarity is just referring to like a cell's ability, or like a property of having like top and bottom have directionality. And this other stuff, the construction of the epithelial cell to cell junctions, because they're trying to move away and become like solo adventurers, so they don't want to get tied in with other cells. Cell shape change, downregulation of epithelial markers, upregulation of mesenchymal protein, secretion of specific proteases, and the reason why we are secreting proteases, an enzyme that breaks down a protein, because we want to break down the extracellular matrix ECM, that's like holding the cancer cells down into places, and we're trying to move around, increase cell protrusion and motility. Okay. Going back to the questions of why do we even have a gene that enables the EMT, the transition from epithelial to mesenchymal, and we can find one of the example here when we're just like on the fertilized egg and to become a baby. We have just this flap out like skin, so composed of epithelial cells, and we form this like round tube that becomes our brain and like a main like a nervous system. So we're going from like a skin flap to a nerve tube, and skin flap it's epithelial, and their neurons are like they derive from the mesenchymal cells. So we need this epithelial to mesenchymal transition when we're trying to develop in the embryonic. When we're embryo. And other examples we need is, besides the development, we also need it for wound healing. When our like skins are like damaged, we need our epithelial cells to turn into mesenchymal cells to be able to move to the wounded area. And another thing is that cancer invasion is, what we're going to be talking about the whole time. So EMT. So what they did here is they activated the EMT gene and see what happened, and what this relative mRNA, what this mRNA levels are showing is so this ECAD, which is here downregulation of epithelial markers such as ECAD, so cadherin is the epithelial markers and N-cadherin is the mesenchymal. So this ECAD epithelial are like downregulated, whereas this NCAD are like upregulated, and these are also marker for mesenchymal. But we can see that once EMT is like turned on, genes are turned on, we see a higher level of mesenchymal marker and lower level of epithelial marker, which makes total sense. Similarly here in this figure C, CD24 is a marker for epithelial, the CD44 is the EMT marker. And we see that when these are activated, these genes, they all move towards to like this way. So the decrease in epithelial, because they're moving to the left, but they have increased EMT expression. So stroma means a support structure of tissue. So we have this tumor stroma, or the connective tissue, and they get this like ECAD, which is the tumor suppressor, and they get more of the EMT transcription factors like twist, as we have seen over here, and then N-cadherin, which is a nerve cadherin, so they get upregulated. So they're oncogenes. E-cadherin is a tumor suppressor, and they promotes EMT. And mesenchymal-like tumor cells release MMPs, which is a proteases, and break down like other stuff, a protein that's holding them down. And so moving on. So CAM, the cell adhesion molecules, there are a bunch of them. They allows the cell to connect cell to cell, and to also connect cell to ECM. And what they do, so what CAM does is they are like a support structure, but also they enables the communication between cells. So one of the things we have is D-integrin receptor, which connects cell and the ECM, and we have a bunch of different pairs. So this CTC, CAM, integrin proteases, moving on, TF, transcription factor contribute to every stage of metastasis. So this is not really a simple thing. It's a complex process. A lot of different transcription factors are involved in the metastasis. And we know there are a lot of different transcription factors here that's involved in the EMT. So intravasation, so going into the blood vessels so from cancer stem cells, when they go in, they become mesenchymal, and then they go through the intravasation process and become a circulating tumor cells once they're in the vessel. So to talk more about the intravasation, there are cells that actually goes in and out of the vessels, and that's an immune cells. So for here we are shown a macrophage. So tumor cells, they kind of like tag along the macrophage to go into the blood vessels. And invatopedia, which means invading foot, they're like basically putting their foot in the door and shove themselves in. And this triangle, this yellow triangle, is showing the interaction of epithelial tumor cells and the macrophage of opening the door on the vessel. So this T-man, which is tumor microenvironment of metastasis. So they shove this foot in here, and then they leave the door open, and then like other tumor cells can like follow them into the vessels and become CTC. That's cancer, circulating tumor cells. Once they're in the, so now we're talking about the transport phase. When the cancer cell goes into the blood vessel, there's a lot of different, it's not a really good environment for them. There's a shear stress from just the blood flowing, and we have different immune cells, and natural killer cells and neutrophils that actively kills the cancer cells, CTC. And one thing that helps them to survive is the platelet. If they cover themselves in platelet, it helps them to stay alive from the immune cells. But in general, it's really not a great environment for cancer cells. And another thing to note is the bloodstream is only going like one direction, so they can't really go back to the primary tumor just because it's hostile. Anyway, because of the harsh environment, the survival rate of the tumor cells is like 1 in 10,000. But because that also means that if their tumors are releasing like 10,000 cells, so like every day, tumor cells to go through, it basically guarantees the metastasis. And it kind of relates to the proximity hypothesis, the distance, the closer distance is preferred to metastasize. So how the breast cancers are more likely to go to lungs because or colon and pancreas to the liver, because they have a direct path to those organs. But beside the blood vessel, we can also travel with lymph, nerves, outside of the blood vessels and through different body cavities. And exploiting this process of metastasis, they're trying to see if they can like detect or diagnose cancer from just drawing blood and detecting the CTC, which gives them advantage of being less invasive and faster. So now we're talking about the extravasations, which is getting out of the vessels, and they go through this complicated process, but they're a little different. Not just the besides the direction of how it happens, it's also the entry of the blood vessel would have been a young vessel, right? Because the tumor promotes angiogenesis, the formation of new vessels. So when they're trying to go into a vessel, it's going to be a new one. But when they're trying to exit out, it's going to be a mature vessel because they have already been there. It's a little different. And the seed and soil theory, which is the theory that explains the organotropism, it's cancer's preference of organs to metastasize. So so different organs have different type of vessels, and different protein expression or molecules on their surfaces. So some circulating tumor cells may have easier time getting out of the vessels than other in different sites. And it's an interaction. So this process of interaction of tumor cells, endothelial cells, platelets, and immune cells, and different ways of extravasations or other ways. So induction of the necroptosis. So they're just like inducing the necrosis of the blood vessels. Another way is when the tumor cells are basically like stuck in one of the capillary, they just start growing and growing to a point they just burst the capillary. So that's called vasculature rupture due to the tumor cell proliferation death trap. So we talked about extravasation, and now we're going to metastatic colonization. And in here, we have to go through the opposite process from going from mesenchymal to epithelial cells in order for them to start proliferating. So they're going from the circulating tumor cells into the cancer stem cells again. And successful colonization also requires a suitable microenvironment. So they need a good environment to be able to start thriving again. But they may or may not start like right away, or they may take some time to just reactivate again. So they may just remain dormant. And we call this term micrometastases. And recently they found out that there's a new role of exosome. Exosome is just a small vesicle that contains different stuff like DNA, RNA, protein, etc. And what they do is the tumor cells release exosome, and they go to some places that their tumor go to places that their tumor CTC will get to. And then once they get there, it prepares a place to be more hostile for them, including the stromal cell reprogramming, cell recruitment, ECM remodeling, inflammation, etc. So they're preparing the sites to be more hospitable before they actually move. And it's comparable to like calling someone to, hey, like before you go to the friend's house, like, hey, prep the dinner for me before I get there tonight. And then once you get there, it'll be a nicer environment because you'll have food. So the drugs that targets the metastasis has not been so successful because we don't know enough about the basic science of metastasis. That's why drugs are keep failing. But things we could do is, so MMP, which is a proteases that's used to loosen tissue, so we can make an antibody that binds to it to decrease the proteases activity, so it doesn't can't move around. It's still stuck with because of the ECM. NM23 is a metastasis suppressor gene, so we can make a drug that activates this NM23 metastasis suppressor gene.